introduction. And as you mentioned, we divided our presentation into 3 parts. I will start with the short introduction of our laboratory. As you mentioned, we are just part of Biological Center of Academy of Sciences. And you see here our campus, which is shared by the University of South Bohemia and also Academy of Sciences. Our laboratory is placed here in the corner. It's shown by yellow arrow. And we are from the town of České Budějovice, which is best known for the Budvar brewery. And as Johanna mentioned, we are part of Euro-BioImaging, and also from 2021, we are also a member of Euro-BioImaging as a part of the Prague node. Our laboratory provides electron microscopy services for more than 30 years, so quite long history. We are equipped to transmission and to scanning electron microscopes. That allow us to just provide quite broad range of imaging technologies for our users, and we are able to just help to image ultrastructure topography at room temperature, also frozen hydrated samples. We do 3D imaging, image analysis, localization of molecules. And I would like to announce that next year, we will have a new microscope, scanning electron microscope, which will have a unique combination of cryo attachment and integrated Raman confocal microscopy and EDS. And it allows us to add chemical and elemental analysis to structures. And we hope it will be powerful and helpful imaging tools for our users. So our laboratory has about 100 unique users each year, and the images that were created by our microscopes appear in 30-40 publications each year, which I think is quite nice. We organize courses each year. This year we held a workshop for advanced users of MIB and AMIRA software, and we received quite good feedback. So it encouraged us to organize the course again in the spring 2006. And this course will focus again it will be for just more advanced users, and we will again focus on MIB, deep learning tools for image segmentation, and also probably for this time with Empanada, because the participation accepted already again Elia Belevic from University of Helsinki, also Kedar Narayan and Michel Darrow. So it will be spring 2006. However, next year we are organizing the practical course for beginners, for those who wanted to learn how to prepare biological specimen for both transmission and scanning electron microscopy. In addition to services, we also are involved in methodological development. We collaborate with the companies like Thermo Fisher Scientific, ContiPro, producer of Hyaluronan, and Kritur, which is supplier of scientific detectors. And I would like to highlight 2 of our results. The first is that we provide standardized samples for cerebral phase skin electron microscopy for Thermo Fisher Scientific. And the second is that our team was involved in development of novel resin with high resistance to charging, which is mainly intended for cerebral phase skin electron microscopy. That's all from me, and now I would like to stop sharing and just give the floor to Jirka to continue with his imaging part.
Hello, everyone. Do you see my screen now? Yeah,
Yes.
so I'll tell you about the imaging that we do in Volumium in our laboratory. This is what ChatGPT thinks it looks like. In reality, it's not that bad, but it's slightly different. So there are 3 main techniques in the SEM, and there are also a couple of techniques in the TM. So the one I will be talking about, the one that we are doing here, it's all started with a sample preparation, like for the transmission electron microscopes. You prepare the sample, stain it, and embed it in the resin. There are ways around it that you can do it in cryo, at least partially, what we do. And then in transmission, you have the electron tomography that we also do here, but that I won't be talking about today. We have the serial block phase, which means that you are getting the sections by having the ultramicrotom inside of the chamber of the microscope. So you have the diamondite that cuts the sample, you image it, it cuts again, you image. The other possibility is focused ion beam, so you don't have the knife in there, but you have the focused ion beam, and you are using that for milling the layers in your sample. So that's available through our node as well, but not in our lab, so I won't be talking about that. And the last thing that we do here is the array tomography. That means that you collect the sections first with the normal table ultramicrotom. You collect them on some support, silicon wafer or the glass, and then you put these sections inside of the microscope and you image it one by one. And then you end up with stack of the images which you can work with. Mostly for the serial block phase, we are using the protocol based on HUA, published in 2015. We generally do some modifications, so unless we really work with the whole mouse, we don't use the perfusion, but the immersion protocol. We extended the staining times for the thiocarbohydrazide and osmium, and we don't use ethanol. We just, for the dehydration, do everything in the acetone, and then mostly we are using the harder plus resin 812, but sometimes we do use it depends on the sample that what we need. That's for the serial block case. For the array tomography, or when we do the high pressure freezing and then free substitution, then we can't use the potassium ferrocyanide because it's not soluble in the acetone, and thiocarbohydrate is also not very well soluble. So we do keep these. We try to experiment with the tannic acid and stuff, but it works with some samples, but not with everything. And for the array tomography, where you usually don't need the staining, so you can use this protocol as well. We can skip it because you don't need everything. So there, the transmission electron microscope protocol just works fine. So just with the osmium and uranyl acetate and lead aspartate. So either you can do it in block, or we do also the post staining of the sections. The problem we face, I think every lab doing the volume SEM, that we try to get, or people want the best resolution, the largest imaged area, and you need to find your region of interest. And that goes against imaging time, because we generally don't want to spend the whole year or a couple of months on every sample. So this is the example of one of our serial block phase runs. So here you can see the block, and where we were, here we were tracking the damage to the brain caused by tick-borne encephalitis virus, and we wanted to see if there is some change to the endothelial cells and cells of the blood-brain barrier. So there were 5 capillaries, so we were having 5 different regions of interest that we were acquiring simultaneously. You have just this part I'm showing here. So here, this is the example of the capillary. There are the erythrocyte, endothelial cells, so on. There is already a lot of damage. So you can see the empty spaces that you won't see in the healthy tissue. There are the viral factories all around. There is already one endothelial cell damage. There is just the stack of the images that we got. You can see that this is kind of intersection of the capillaries. One is going from the top, one is going from the left. They join and then continue towards the right bottom, or we don't know where the flow goes, right? So we can't tell. But so this is the example. The field of view in the serial block phase, is the advantages that can be quite large. So this is the example of unicellular, of the cell culture of the unicellular organism, Trypanosoma brucei, and the advantages that you can, in one run, collect a lot of the cells at the same time. So if you compare it to the FIP, which is really popular and which has much better resolution, at least the gallium FIP, there you will get much nicer resolution, especially in Z, but generally just for one cell. So I think the best is to combine having one cell with a really good resolution and then have a whole bunch of other cells where you can tell that all the cells are actually the same looking as the FIP one or not. This is the example of the one cell cropped out of that data set. And this is to show you that the volume SEM is really great in quantitative thing, that you can quantify everything that you have there, you segment. So this one particular cell has volume of 77 micrometer cubic. Its nucleus is 5.5 micrometers cubic. The mitochondria is 12 micrometer cubic. We can measure the length of the flagellum. We can measure the length of the cell, etc. And what you can use it for, you can actually use it for distinguishing the phenotypes, either knockdowns or anything. And in this case, the group was over-expressing a viral protein. And normally you would have the mitochondria just one single tubule. But here you can see that after overexpression of this protein, there were some big pockets of cytoplasm trapped inside of the mitochondria with no communication with cytoplasm. So you won't see it unless you would use the volume SEM. Because of the large field of view, you can compare also the even pieces of the tissue. So this is what healthy lungs look like. You see a lot of alveolar space where the air is, so you can breathe. And here you see after the COVID infection that there is a lot of immune infiltrate. So there is no surprise that people having lungs like this won't be able to breathe. Because of the large field of view, you can find like rare events. So in this case, you can see this is pneumocyte 2 with the COVID particles inside of the cell. And here on the left side is the alveolar macrophages that are actually going from one alveolus to another through the septum. So in blue, you have the septum, and there are two alveolar macrophages. So the macrophages can actually kind of dissolve the tissue at the tight junctions, which are here marked red, and they will go through the tissue to where they want or where they need, or where, in this case, where the disease is or where the virus. So what we need to do is to find, unless you have the cellular culture or a pellet, you need to find your region of interest. So we did use, when your region of interest on the surface, we did use the minimal resin embedding method. It was originally published for the PIP in EMBL in Heidelberg. So we modified it for the use in the serial block phase. You can see that you can actually nicely see the surface and all the details on the fish roe. Here, this is the excretory organ of the springtail. So when you have it, you can actually see how much or what's protruding above what you need to trim before the imaging. But we found that this minimal resin doesn't only help you with the finding your region of interest, but it also helps with the charging. Charging is one of the biggest problem in the serial block phase. And you can see when your sample is inside of the resin, which is here and there, then the empty resin around actually function as an insulation layer. So the charge cannot dissipate and it actually distorts your image. So when we remove this resin, we're actually able to image these samples in much better conditions in low vacuum or even or mostly in the high vacuum. So it was a big difference. And because the imaging condition was much better, we're able to even use the multi-beam deconvolution. So normally the physical limit for cutting with a knife is around 25 nanometers. That's what the people achieved in the past. So here with the multi-beam deconvolution, we are able to even achieve like isometric voxels, like 10 times 10 times, or even 8 times 8 times 10, which for a serial block phase, I would say it's really good. So when we can't use the minimal resin, then you need to kind of know what your sample is and what it looks like. So here we are looking for the digestive duct of the tick, and people in the lab like Maria are able to tell you that this is actually a start of the synganglion. And we know that through that synganglion is actually the digestive tract going through. So we used it the serial block phase, like for higher expensive microtome to get through to the place where we want it. It took us almost 2 weeks because we couldn't run it overnight. So we have to like watch where we are. So now when we know it's much deeper, we would go there much quicker. And when we get to the, so we were imaging at low resolution. And when you get into the place where you want to be, you can image in higher details your region of interest. So I was always showing that you can acquire like multiple region of interest in better resolution. The ROI can be actually followed. So you can change and move the ROI when the cut moves around. And the power of the 3D is when you look at in the intestine here, there is a lot of dots. And if you have just one line, you won't be able to tell what is what. But if you have a stack of images, you can actually connect these dots. And you realize that these dots are actually the spirochete bacteria. So you're able to find the bacteria inside of the tick. When it's possible, and we can use it, we are using the PLEM, so the fluorescence for navigation. So this work I will be showing is done with the collaboration with Pablo Suarez from Max Planck Institute in Berlin. And he got actually the Isidore support from the Euro-BioImaging, and he needs to find the Plasmodium falciparum inside of the mosquito gut. So this is the mosquito intestine, so you can image how small that thing is. So we use the standard TM grid for the coordination of it on XY. We use the motorized stage to measure the height in Z. You can actually see the little dots here because the resolution is not enough. But the parasites were located in this place, so we made the pyramid at this place. And we tried the serial block phase at the beginning, and we actually managed to image 1 out of 3 parasites. But it turned out that the serial block phase, because there is the limitation with the electron dose, we are not able to get sufficient resolution or what we needed, because the Plasmodium falciparum is really small, the cells are small, and the organelles that Pablo need to see are really tiny. So we have to switch for the array tomography. So we first collect the sections. On the sections, you actually find your parasite. And there, this is actually the Plasmodium falciparum inside of the mosquito cut. So what Pablo does, he actually has a knockdown of the chest that is part of the excretory organelles of the Plasmodium. And the excretory organelles are used by the parasite to traverse through the host tissue. So there are like different kind of the organelles in different stages. And Pablo, when he knocked down this component, he found that the parasites it looks like they are not able to get through the tissue. So he wanted to see what actually happens to the organelles. So that's what we did. There was a lot of different samples. There's a lot of different stages. Some of them we don't actually need to find in the mosquito because Pablo is able to purify them. So we get them in the pellet, but still the amount of the material is quite small, and especially the sporozoites are really even tinier than the other stages. So it's really hard to image. So here you can see for the OCAGNet, we still didn't finish the analysis, but all the stages, the results are pretty similar. So on the first line, there is the number of the excretory organelles, and here is the volume. You can see that in wild type, upon the activation, so the activation is done by increasing temperature, and activation means that the parasite actually excretes the organelles. So you can see that in wild type, after activation, the organelles are almost not detectable because they excreted their matrix or whatever was in there, and you don't actually see them anymore. But after in the knockdown parasites, they were not able to excrete fully or properly the inner parts of the organ. So that would be the reason why they are actually not able to get through the tissue. And that's potentially very interesting for the treatment. So this is just for repetition. Again, like how we did it in this case. So we use the grid motorized stage, and sometimes the sample is too big for the grid, but there is another way how we do it. So in Borrelia, this is again in the tick. So tick is big, and there is so we use the tissue as a marking of the place. So you see actually with the fluorescence where the Borrelias are. You can map this region. So after embedding the tick in the resin, you can, with the tool in blue, see the context, see the different tissues, and see that in this place, this is actually the base here. So this is what we did with the stereo block phase, because the resolution and imaging was enough. So we made a pyramid here. So this is the block phase of the pyramid. Here in the detail, you can see this is actually the cut where the Borrelias are. And this is the, this is the reconstruction. Well, I can't run the video. No. So well, I will not run the video. But you can see that in this case, the Borrelias are actually traversing the tissue in between these two cells. So there is the yellow cell and the pink cell, and the Borrelias are in between. Can we get some other information than just the structure or the structure of the sample? So what we try to do that as well. So we were imaging the diplonemid species for Daria and others, and they found out that in this species on the seal block and the transmission, there was some weird organelle. So it was a bunch of vesicles and stuck of the tubules, and it was never really seen in any other organism. And the hypothesis was that it has a role in endocytosis because it was generally close to the cytopharynx where the endocytosis starts. So to address this, we try to feed them with some electron-dense material. Normally, the gold, nanogold is used, but the nanogold was precipitating, probably because these are the saltwater ones. So in saltwater, it doesn't work. It's pain to work with these because they are really sensitive, and we have to high pressure freeze them, and the staining doesn't work very well. But you can see that, so we actually at the end we use lanthanoids that we are using for the cathodoluminescence because it emits light under the electron beam, so you can use it also for tracking some other things. And so this is the images of the transmission electron microscope. Here you can see the organelles where it end up. On the video, it was actually also in some other organelles of these. So we used array tomography because there is less stain, so we get higher contrast from the lanthanoids. And what Mario already said, that we want to get the new microscopes. We did pilot studies like within the collaboration. So again, Daria and Jana Pilato, they found that in some other diplonemid species, there is this big, on the Raman, there are like big concentration of strontium and barium, which is kind of weird because you have the strontium and barium in the seawater, but the concentrations are super low. And these organisms are kind of able to purify it and store these things. So we saw the crystals in the TM and serial block phase, and to prove that these really are the strontium crystals, we cut thicker sections for the array tomography, and we send it over for the EDX mapping. So here you can see there is the crystal. So this is the region. The general mapping, you can see that the osmium is pretty much everywhere, while the strontium really comes up from the crystal. And with this, I will finish my presentation, and Fanda will continue with the software.
Here. Hello, thank you, Erika, for introducing me. And this is the last part of the presentation focused basically on the processing and analysis of the volume EM data. I will start today with a little bit of history because this is the biggest and oldest project we did regarding the volume EM, and it was the manually acquired array tomography which was really without any software, specific software aid and without any specific software aid for the reconstruction, hand-drawn manually to reconstruct the feeding mechanism of the tick. And from this, actually, this moving model of the tick feeding was being reconstructed. And yeah, this is the oldest one I know about. So it's in 2017. But now, of course, the pipelines are looking a little bit different. First, we start with the acquired data. In our lab, as Jirka already mentioned, we work with the SBF-SEM array tomography and electron tomography, which we are capable of recording ourselves. But sometimes we are provided with some FIB-SEM data and also rarely like soft X-ray tomography, which can be processed in very similar manner. So the next step in this data processing is, of course, stitching and alignment, which is in our case mostly done in MAPS, which is a software provided to our upper volume scope from Thermo Fisher Scientific. However, if there are some minor issues, we use it or we do it in the these processes not in the maps, but rather in Fiji and module TrackEM. And when we need to do some very fine alignments in the very end during the segmentation, we use the MIB microscopy image browser. I will be returning to the software quite a lot. So the next step in the data processing pipeline is filtering and binning and bit depth conversion. This is again in our lab done mostly in MIB because it's also batch processing compatible, and it means that even if you have a big data set and not as powerful computer, what you can actually do is that you set up the pipeline and start it overnight, which is really nice, and you come in the morning and the job is done. So after this pre-processing step, these steps we get to the image segmentation. Again, mostly in our case done in MIB with there is a variety of the manual tools, semi-automatic tools. There is implemented segment anything model. There is also deep learning via the DeepMap. And when in case we work with the electron tomography, I do it in MIB, or my colleague actually does it in iMod, but I'm not working with iMod that much, so I will not mention it anymore. This is the last time. And the last step of the data processing is visualization and can be also quantification. And this is what we do mostly in Amira, the quantification in the MIB as well, but the visualization is mostly in Amira. So I mentioned already MIB software, which is MATLAB-based image processing software or image processing tool developed by Ilya Belevich in a laboratory of AIO Kitalo in Helsinki. And it's very nice because it exists in two versions: standalone, where you don't need the MATLAB license if you are researchers, and the MATLAB version where you need installed MATLAB and specific toolboxes to actually run it. It's good to be a student, by the way. So when you are working with the MIB, you can do the pre-processing step, which can start with the alignment. You can filter the image there. You can bin it. You can also decrease the bit depth. And after these pre-processing steps are done, you go to the actual segmentation. Again, I will mention some manual tools, some semi-automatic tools, then the segment anything and deep learning as well. And after you are done with these, you can go also, you can use the statistics to get some more quantitative information. And one of the specific use cases is also stereology. I will mention it. We use it differently than it's offered in MIB, but it's still doable and it's really nice. So the first time we encountered this software was actually, or I encountered this software was in 2019. And the portfolio of tools for my use by that time was pretty low because I didn't know how to use much of them. And I started with the brush, which is really useful tool for some overall selection. And I think everybody who has a Microsoft Paint knows how this tool works. Then the second thing is the membrane click tracker, which is a really nice tool for tracing membranes. And for example, when Jirka showed you showed you the macrophage going through the cell. It was the tight junctions were traced exactly with this two, because it's following the similar or as close pixel intensity as possible. And the last one, which is all-time favorite for the light microscopists, is the black-white threshold, which is really nice to select some highly contrasting organelles. It works both in 2D and 3D, and you can also do it in global, which means on the whole data set, or you can just pre-select an area where you do the thresholding in. And these tools are sometimes a little bit slow, and but they are still sufficient for a specific application. But if you want to scale it up, it bites you quite heavily, because, for example, one trypanosoma with these tools is fine, but 40 of them, it's work for like 7 to 9 months approximately. So let's go to something a little bit different. This specific application will use only the brush. And this was in 2020 when the COVID-19 actually appeared and started to be solved. So we recorded the data sets of the COVID-infected lungs on the humanized mice. And we recorded three data sets with 600 gigabytes in total and about 4,000 sections. And if we wanted to segment this manually only with the tools I mentioned before, we wouldn't be done still. So we decided to go a different way, and we decided to go with the stereology path. So what the stereology is and which principle we are using, it's Cavalier principle. It starts with the image, where you know the dimensions and the pixel size or voxel size. Then you apply a stereology grid with known sampling steps. In this case, it was like 2 by 2 by 5 microns in the voxel. And then on each cross of this grid, you actually annotate, label, however you want to call it. You annotate or label what structure is actually underneath. In this case, you can see, for example, the yellow one here. For example, you can see here the erythrocytes, you can see the capillary, you can see the empty resin, and so on and so forth. So after you are done with this coloring or annotation, you open the statistics tool. Okay, yeah. You open the statistics tool, and the only thing you are interested in is actually the number of the dots. Nothing more than the number of the dots, which in this case was, for example, 45. And then you put all these numbers, you add them together and put them to the table. And from this, you can pretty easily calculate relative volumes and then to compare them. How does it look like in the very end? For example, we have the data set with no infection, data set 2 days post-infection, data set 5 days post-infection with COVID-19. And you can see that the alveolar space is decreasing from the control non-infection to the 5 days. It also, the count or the volume of the alveolar macrophages is increasing. Also, the overall volume of the immune cells increase in the necrotic tissue, in fibroblasts. And for example, the endothelial cells volume is decreasing with the advance of the infection. So this is really nice and fast way how to extract a lot of quantitative data. You cannot do any modeling out of this, you cannot render it out, but the tables are usually for scientists more important than the nice renders. So now let's go to some bigger project with, again, a real segmentation. And it was the segmentation with TIG-GAT, which was done in end of 2022 and the beginning of 2023. And this dataset was rather big and would be even bigger if we haven't decided to go with quite lower resolution, because we went 15 by 15 nanometers in X and Y and 2080 in Z, which means we took only every 4th section. We cut it. And we wanted to get somewhere here. And of course, to select these different organelles and parts of the tissue, it had to be segmented on its separate way. So, for example, the lumen of the TIG-GAT was really well segmentable with the superpixel brush, which means we are using the brush, but instead of using pixels for the selection, we are pre-calculating the dataset to the superpixels. And then we have pretty nice guide, and the segmentation is way faster and way more precise. And also this was compatible with the tool GraphCut, which uses these superpixels and supervoxels. Similar approach was employed during the segmentation of the whole tissue, where we selected just the outside of the cell at some area, and then flipped the selection or inverted the selection, and then used this small area for training and for the deep learning training. Then we predicted it on the rest of the data set, and we manually polished whatever was wrong. The next part of the data set here are the nuclei, which were again segmented with the facilitated brush or with enhanced brush with watershed superpixels. In this case, then we used the deep learning again, because there was a really huge number of the nuclei patches. So we segmented just few of them, and then again did the deep learning and predicted and polished everything. The next part were the vesicles. These were quite tricky because simple threshold wouldn't work for them, because as you can see, you have two different or various shades of gray inside of them. So in this case, we just needed to approximate a bit. We selected just the brighter parts and then filled everything in. And again, we had to polish it and then use it as a training around like 30 sections. And we use it as a training for the deep learning network. We predicted everything, polished it manually, and went further. Then there are not only one type of the vesicles we are interested in, but another one which had a different shade of gray, but did not actually fit to, but were located only close to the lumen, which meant that we could use another technique, use the black-white threshold in expanded mask of the lumen, which is sounds a bit more complicated, but in the end the job was pretty easy. Again, then we did the polishing of this rough automatic selection, and did the deep learning and prediction for the rest. And the last part, which will be visible in the final model, were dark vesicles, which were completely black, and they were really easy to select because we could just do the black-white threshold within the eroded mask of the whole tissue. Okay, so now, sorry. Now we decided to combine all the individual segmentations and polish it. But let me get back slightly to the deep learning, because as you can see here, this is the training data set where we use just the black-white threshold and manual polishing. And then this is the prediction. It looks pretty nice. It looks pretty accurate as a matter of fact. And if you look at the numbers, it looks even better, because the prediction was pretty accurate. It was like 97.3% after a couple of hours. However, on the next slide, you will see what could go wrong and what went wrong, actually. And this was that we did the training and the selection only in the central part, where the thresholding worked pretty nice and pretty smoothly. But when we tried to predict everywhere on the whole data set, you can see a lot of areas which were actually, zoom in somewhere. You can see here that there is a lot of areas which are not the vesicles, and we still had to somehow count for them. So the pathway here was that we just cut this area, polished in this area again only the vesicles, and train the network again, and then run the prediction, and then everything was way better. It was not perfect, but it was way better. And now here you can see how the model look in static point of view, and here you can see how the actual model of the TIGGAT look. It was running all the time I was talking about the problems with the deep learning. So here you can see the last individual cell, where you can see the nucleus, the lipid vesicles. the hemoglobin vesicles, the lumen, and yeah. And then later of 2023 came, and Meta decided to release their segment anything model, which is really nice deep learning model used for image segmentation, and it's implemented as AI script. It's a Python script, and it was implemented pretty fast in a lot of softwares. In Napari, which is Python, it was in May. In MIB, which is MATLAB, it was already in April. And I was quite lucky that I was capable of having it in my computer. And then it later appeared also in ImageJ already this year. And then you can see that, for example, the paid software such as Aravis Cloud should have it implemented in the next big update, because it's still being worked on. And now, what is the best part on it, that the network was not only released, but developed. And now, for example, the segment anything 2.1 is really, really fast and really user-friendly, as you can see here. If I had this, or if we had this tool in possession when we started the segmentation, this is how it would probably look like, the preparation of the training data set. This is the tissue. This is the lumen. This is the lumen, and this is the lumen. Now the nuclei. Sorry, this is a GIF, I cannot skip it. So these are the nuclei, and everything is just one click. No super pixel clustering, nothing. Only it's quite GPU heavy. That's the only downside on it. But I think the speed is like giving or balancing some of the cons, but this is like really, really fast. You can see that even the vesicles could be segmented like this pretty fast and pretty straightforward. So then, of course, once we learn to use this this tool, we could do way more data, way faster. The data on the left are showing the viruses and viroplasm in the infected tissue. This was the data set done in collaboration with Eva Durinová Ramantuma, and they were imaged by Javier Conesa from Madrid. And these were, for example, segmented with segment anything and black-white threshold, which generated from 400 images, like about 20 of them for the training data set in a couple of minutes. Then it ran a couple of hours for the training without my presence. And this is basically the result, how it was predicted, of course, after some polishing. And I have to say that the final polishing and the animation of this was done by my colleague Tomáš. On the next one, which was moving here, was the Borrelia interaction with the capillary. You can see the green endothel, the blue pericyte, and again the tight junctions, which are again quite contrasting, so they are pretty easily to be segmented with the click tracker. But in this case, if I remember, some of them were selected by the brush as well. And the last one is the aforementioned soft X-ray tomography, which was done in collaboration with Jana Pilatová and Caroline Larabel from USA. And again, this used just segment anything, brush and thresholding for about 450 sections, and we have way more data sets for this. So yeah, this was everything from my side. Thank you for your attention, also for my colleagues. And I also need to acknowledge a lot of people, and I think I will not name them because I think we wouldn't be done by the Christmas Eve. Thank you.